All right, we're going to be looking at transcription and gene expression. In this screencast, we're especially going to be looking at some basics of transcription that we need to know at the HL level, and then we're going to be looking at how we can regulate the expression of genes in a few different ways. So first, just kind of one of the basic HL things that you need to know is that transcription is going to be happening in the 5' to 3' direction. So the new mRNA molecule that's being formed, notice you can only add the nucleotides to the 3' end because there's no phosphate there. And so these get added to the 3' end, so it's growing from 5' to 3' in direction, just like in replication when we're copying DNA. All right, so now the focus of gene expression. So there's a really nice diagram that I have posted in this lesson plan that you can take a look at that has some details about all the different ways we can control which genes are being turned into proteins and how along the way, out of all the steps of protein synthesis, we can control those things. So the ones we're going to focus on in the beginning are here at the chromatin level, at the transcription level, and then with RNA processing once that RNA has been formed. So these are kind of the beginning. There's still more once we get down to translation. We'll be doing that later on. So first of all, we can regulate transcription by, making sure that chromatin is superpacked or not, so we can modify the nucleosomes, and also with transcription factors that can help determine whether a gene is going to be transcribed or not, allowing that RNA polymerase to attach. Then once transcriptions happen, we can do it at this RNA processing level, which is modification of mRNA. So there's different ways that mRNA can be spliced and put together that can result in a different protein. So we're going to look at that in detail in another screencast. And then finally, the environmental impact on gene expression. So at the environmental level, all of this can be affected as well. All right, so we're going to dig into these first couple, the regulating transcription with modifying nucleosomes and transcription factors. So here's just kind of in general, here is the essentially gene unit. So here's where this is going to be like the protein, the DNA sequence where the protein would be built, but there's all these other parts that help with transcription of turning that into mRNA. And so we're going to take a look at how this stuff upstream from the gene itself can help control that. And then you see once we make the mRNA, then that can be cut and spliced in different ways. So you get a final mRNA that could be a different protein than another. Okay, looking up close. First of all, this promoter region, which you see here, there's this promoter region. We've talked about that as well. So this promoter region is where the RNA polymerase will attach to the DNA. And it needs to be attached for transcription to happen because we know that it's going to be the one catalyzing the formation of the mRNA. So this promoter region is important because this is actually where the RNA polymerase is going to attach. And that is another example of non-coding DNA that actually has a function. So we don't actually make a protein out of here. You see transcription doesn't start until it gets to the gene, but it's imperative for that RNA polymerase to attach. So it doesn't make, the nucleotide sequence doesn't translate into an amino acid sequence ever. However, it's imperative for transcription to happen. So it has a function, even though it doesn't code for amino acids eventually. Okay, so let's take a look at this, this upstream area. So you're going to see there's a lot of different ways that we can kind of help that RNA polymerase attach or not. So first of all, you can see that there's the promoter region, but sometimes there's these other parts that are like way far away. You don't need to know all the names of these, but these other parts upstream can help the DNA polymer or RNA polymerase attach as well. So let's take a look at what that might be. So often RNA polymerase is not going to just go up and attach on its own to the promoter region. There's generally a number of proteins, regulatory proteins. Another name for them, for these specific regulatory proteins, are transcription factors. And these proteins help the RNA polymerase to start transcribing. Okay, so Without these transcription factors or these regulatory proteins, the RNA polymerase isn't going to easily attach to the promoter region and begin transcription. So these transcription factors are usually proteins, these regulatory proteins that maybe were made on another gene somewhere else. So they have to be present. If they're not present, then transcription is not going to happen. So we can regulate the presence of these regulatory proteins by transcribing and having those available or not. Also, there's the enhancer, these areas that I was talking about. The DNA can actually fold over and help stabilize the attachment of the RNA polymerase. And so these proteins can help, with that and these upstream areas of DNA as well. And there's proteins that act as activators that can then get it to fold over and help with that stabilization. Okay, so besides the fact that we need the polymerase to attach, the RNA polymerase to attach to the promoter region, which can be regulated by proteins, right? Regulatory proteins. If those proteins are there, it can happen. If they're not there, it's not going to happen. But another way we can regulate it, actually prior to ever the DNA polymerase attaching, is that with the nucleosomes and some of the DNA, we can attach these extra molecules to them to allow the DNA to be more or less packed. So if DNA is super tightly packed, right, with the nucleosomes, RNA polymerase cannot bind to the promoter region. So we often need the areas that we want to be transcribed to be loosened up a little bit to allow the RNA polymerase to attach to the DNA. When it's all packed like that, the RNA polymerase can't get in there and actually transcribe the gene. So the histones will play a role in that. And what's happening here is that, remember all the tails that are hanging off of the histones? So here's the nucleosome, right? The nucleosome is the 8 histones plus the DNA wrapped around it. So the histones, which are the proteins in the nucleosomes, have little tails, and they can be chemically modified. And if you add an acetyl group, so these little acetyl groups, to the histones, that actually forces the DNA to unpack a little bit, and then it stretches it out. So acetylation of histones actually promotes the loosening up of the chromatin structure, and it's going to allow transcription because now the regulatory proteins and the RNA polymerase can attach to the promoter region and get in there and start transcribing the gene. So acetylation actually can allow transcription to occur. Okay, it promotes transcription. There's another chemical modification, but this happens actually to cytosine nucleotides. And that is called methylation. So if you add a chemical called a methyl group, what you're doing is you're methylating cytosine nucleotides on the DNA, and that causes it to tightly pack. So this is highly methylated where it's red. So there's a bunch of cytosine nucleotides in the gene, and they have a methyl group attached to them, and it causes the nucleosomes to pack super tight. If it's packed super tight, the gene is really just switched off because the gene is packed together, the RNA polymerase can attach, and usually the histones have been deacetylated. Here, when you have the gene switched on, notice that the methyl groups are not attached to the cytosine nucleotides, and that can allow acetylation. It can loosen up the chromosome. Okay, so methylation stops transcription from happening, whereas acetylation promotes it. So methylation stops it, acetylation promotes it, okay, based on how tightly packed that is. And something super interesting about that is that I just wanted to share with you is that, you know how females have 2 X chromosomes? Well, it turns out that in any one particular cell, only one of those X chromosomes actually will be used for transcription. The other one is super highly methylated and is basically inactivated. So only one of those 2 X chromosomes will actually, be available for gene expression, which is going to be really interesting. In gene expression, there's some really cool links to why calico cats are calico. We'll get into that when we're doing genes a little bit later.